Okay, we're going to start again. Welcome back from lunch. I hope everybody had a great lunch and a great opportunity to talk with colleagues and maybe some new people meet some new people in the meeting. That's one of the purposes of these public meetings as well. So as we move, we're going to continue on in this session on federal-state collaboration, and we're going to dig a little deeper, hearing from the Minnesota Department of Health on how they're using whole genome sequencing in their public health practice, and then also how the FDA and NARMS is using whole genome sequencing to look at the public health problem of antimicrobial resistance in bacteria. And lastly, we're going to hear, I guess this whole session was intended to kind of illustrate how federal and state agencies are working together, and that's really going to crystallize in the last presentation where we're going to talk about GenFS, which is an opportunity for agencies to get together and work on these problems of standardization, common protocols, all to improve the quality of the data that's going into the common database at NCBI. So first we're going to hear from Dr. Dave Boxrud from Minnesota and Dr. Patrick McDermott from FDA Center for Veterinary Medicine, and lastly from Dr. Bill Klimke at the National Center for Biotechnology Information. And again, we're going to ask for folks to hold their questions. We are going to have a question and answer period at the end of the day after our break. So, Dave, thank you.
Hi. I'm going to talk about whole genome sequencing at the Minnesota Department of Health. I was asked to talk about how sequencing is being done at the public health laboratories, the state public health laboratories, which is a pretty difficult task because there's 50 states and then there's many local health departments. So I'm going to give a little bit of background about how public health laboratories nationally are incorporating whole genome sequencing, but then really kind of focus on how we're doing it in Minnesota as just one example. So this talk is going to give us a status update on whole genome sequencing at public health laboratories, talk about the role of whole genome sequencing at public health labs, how we communicate whole genome sequencing information at public health labs, a little bit on how we've evaluated whole genome sequencing and so we can understand and interpret the data at public health labs, and then lastly I'll close with an example of the utility of the sequencing. So John Besser showed this slide a little bit earlier, but I wanted to point out that in 2014, the Association of Public Health Laboratories did a survey, and at that time, 21 state public health labs had a sequencer in-house. Now, we have 43 labs in 37 states are certified, which means that they have not only have the sequencer and have the training, but they have been able to show proficiency with their sequencing. In all states, all public health laboratory, all states in the country have been financed for a sequencer, but so by the end of 2017 or early 2018, all states will have a sequencer in-house. But there are still a number of issues that state public health labs are dealing with. Right now we're in this incredible transition time, and we're continuing to do pulse field gel electrophoresis, which is a conventional or traditional subtyping method, while we're transitioning to this new subtyping method. It puts a tremendous resource crunch on the laboratory. So that's a difficult, probably our main challenge right now in public health labs. Many public health labs have IT issues, either with storage or their IT departments don't allow them to use certain types of software. Some of that has been largely resolved, but there are still some issues in some states. Training on both the wet lab side and on the bioinformatics side using the data is still a little bit of an issue, but it's been resolved in a lot of areas. Bioinformatics resources is a little bit of a challenge. John talked about how PulseNet is doing their analysis, and that's going to be very helpful for a lot of public health laboratories. But right now, we're still in a little bit of a waiting mode. And lastly, ordering reagents is a bit of an issue for public health labs. Reagents are very expensive, and sometimes certain states, it's really hard to put 5- or 6-figure orders in, and that causes a lot of problems with their ordering issues. So I just want to start with the very basis of foodborne disease testing. This is traditionally how we've done our testing. And this is, I really think that this is the linchpin of how we identify outbreaks. On the laboratory side, we get human samples in, we analyze the samples and subtype them, subtype the bacteria in them, the Salmonella or E. coli, and then report our results to epidemiology, really focusing on clusters so that they can investigate them. On our epi side, we interview cases and then investigate clusters and lead investigations. And I think what is really important is this process, this work on both sides, but also the relationship between the two. There should be a sense of urgency with all of this work and with how this data is being communicated back and forth. Whoops. And, but of course, we have a lot of other partners that we work with regularly with other state and local health departments, USDA, FDA, CDC, and PulseNet is certainly been a huge partner, and also with our environmental health team in the state for investigations. And just to talk a little bit more about epidemiology information, these interviews that they do, I'm not going to go into a lot of details, but these are really, really important to try to get as good of exposure data as possible. And to get a really good interview takes a lot of time. You're really asking a lot of in-depth questions of what people ate, where they ate, specific brand names, when they ate certain food, restaurants they went to. Each interview takes anywhere between 30 minutes to 60 minutes. And one of the ways that we in Minnesota that we have for routinely sharing subtyping information is we created an automatic daily report through our Laboratory Information Management System, or LIMS system. And it's broken up into a few different pieces, but the idea is that we're able to have a standardized and methodical way of getting information from our laboratory to our epidemiologists. And I'm just going to talk a little bit about a couple of these parts. The first part is just essentially giving a report of what was seen the previous day. Very simple information, some background demographic information on the cases, but also the subtype information. In this case, it's the PFG pattern. So every PFG pattern gets a name so that we can communicate that with our epidemiologist. The next part that is really key is that we take a look back at those subtypes or those serotypes and say, have we seen that for the last 30 days? And that information is also sent to the epidemiologist. So in this case, we had 7 isolates that had the same PFG pattern that we're seeing in the last 30 days. Obviously, this is a cluster that our epidemiologists would be very concerned about. So now that we're going to a sequencing-based method, a lot of things stay the same, and there are a few differences. On the public health lab side, we're still looking at human samples, but instead of doing pulse field, we would be sequencing those samples. But we still need to report that information to our epidemiologists. And from the epi side, they're still doing their normal stuff. They're investigating, interviewing cases, investigating clusters, and looking for outbreaks. We do have some more partners. It's actually the same partners initially, but we have another partner, which is NCBI, which is where we have a sequence data repository. I would also be remiss to say that FDA has been a much bigger partner with us with sequencing. We are a genome tracker lab, so we are sharing data back and forth with FDA, and they've been a tremendous partner for us. So now that we're working with whole genome sequencing data, the communication is a little bit more challenging. Some of the previous speakers talked about nomenclature and how they're naming their patterns. So far to the public health labs, we don't have that available. So we created a number of spreadsheets to give this information back and forth. And essentially, the most important thing is that we have a cluster ID. We tell our epidemiologists what is a cluster, and then they investigate those cases. And we also send them an email that lays out what a cluster is and gives a little bit of background information. In this case, it's 2 cases, same PFG pattern, gives some information. It's very easy for them to understand this and to create action on this. The negative side of this is that this is not going to be sustainable for a large number of organisms. It works in the short term, but it's not possible to work in the long term. So now I want to focus about how we determined what is a cluster and how we have used that information that we learned in the past going forward. So I'm going to talk a little bit about Salmonella enteritidis, a study that we did with Salmonella enteritidis. It's the second most common Salmonella serotype in Minnesota, but it's very clonal. Here's the major pulse field patterns. There's about 5 patterns that make up about 80% of the isolates in Minnesota. So pulse field really does not do good enough job of providing diversity for Salmonella enteritidis. So we decided to do a retrospective study on SE, and we wanted to look at stability. How stable are the sequences over time? Typability. Are we always able to get sequence data from every isolates? Discriminatory power, and also epidemiologic concordance. Epiconcordance is essentially defined as if it's an outbreak, they should look very similar. If they're not related, they should look very different. So our laboratorians and epidemiologists met, and they came up with a study set. And we had used some very well-characterized isolates. We looked at isolates from 7 outbreaks, but also looked at 22 sporadic isolates. We also looked at some in vivo isolates, so multiple isolates from one person over time. And we worked with the New York Department of Health for the analysis for this. And what we saw was that within an outbreak, there were very few SNPs. There was a maximum of 3 SNPs within an outbreak. And when there were sporadic isolates, they looked very different from the outbreak isolates, even when they were the most common PFG patterns. So that provided us some information on how to interpret this data going forward. So then we did a prospective study. Our conclusions were that that the sequences are stable within a person over time. All isolates were able to be typed. There was a lot of diversity, and there was really good epidemiologic concordance. Outbreak isolates looked the same. Isolates that were not epidemiologically related looked very different. So we used this data to do a prospective study, in which case we looked at whole genome sequencing and pulse field gel electrophoresis clusters in real time and tried to identify the source of them. And what we saw was that with sporadic isolates, by whole genome sequencing, they look very different. There's an average of 93 SNPs different between the most common PFG pattern that were sporadic. So that's, going back to what we saw from the outbreak isolates, there were 0 to 3 SNPs different. So from a sequencing perspective, it's quite easy to see what is likely to be related and where we should focus our investigation. And also, the number of clusters that we saw with whole genome sequence compared to pulse field went up dramatically, but the number of isolates within each cluster went down dramatically. So I think this provides us with a great opportunity to identify outbreaks earlier with fewer cases. And lastly, I just want to talk about how we use this data going forward. One great example. So this is a the most common PFG pattern in Minnesota in August and September of 2014, with each box representing one case of the most common PFG pattern. So there were 19 total cases. So by PFG, it's really hard to understand what is going on. We just know that there's a lot of cases. When we did whole genome sequencing, we found that there were 8 of the isolates that were 0 SNPs from each other. So we investigated those to try to understand what the relationship between those cases were. And so our epidemiologists did a great job, and they were interviewing almost all of the cases, and they found that 6 of the 8 all ate a frozen chicken calf product. One of those 8 was a secondary case, and another one of the 8 may have eaten a frozen chicken calf product. They ate a lot of frozen stuff, so they didn't remember it specifically. Of the 11 that were not a part of this cluster by whole genome sequencing, none of them ate this product. So we were able to use the whole genome sequencing to identify a product that would have been very difficult to identify by pulsed field gel electrophoresis, and it certainly would have been delayed. So the future of whole genome sequencing at public health labs is going to continue to increase. WGS will provide more information methodically compared to current methods, and it's very close to replacing some of our current methods, such as pulsed field gel electrophoresis and serotyping. When we have a standardized WGS nomenclature that CDC is producing, that will greatly improve our communication and make the data sharing much easier. So in conclusion, WGS is a tool that can help us identify clusters and outbreaks better than our traditional methods, but there are current challenges, and there will be additional challenges as we implement this technology. But speed and communication will be vital to continue these aspects for outbreak investigation. Thank you.
Good afternoon, everyone. Thank you to USDA for the invitation to join this conversation and show you some of the ways in which we're exploiting this very powerful next-gen sequencing data in our antibiotic resistance surveillance work at FDA. Okay, well, or not. Oh, here we go. Sorry, I don't know what I'm doing with this. Okay, I only had one button left to try. So a little context. I'm sure everyone in this room is well aware of the global threat posed by antimicrobial resistance, and a great deal has been done even in the last few years at the highest political levels to encourage and lead countries in developing, and I should say dedicating resources to addressing it on a global international level. And WHO came out with their Global Action Plan for Antimicrobial Resistance back in 2015. And some of the summary points in that report that help set the context here that they point out is that the development of resistance is linked to how often antibiotics are used. I'd say that's true in nearly every case, not every single case, but because many antibiotics belong to the same class of medicines, resistance to one specific antibiotic can lead to cross resistance to others. And I think if you hear these simple facts, you'll start to see quickly how whole genome sequencing can resolve some of these issues in more detail. Resistance that develops in one organism and location can also spread rapidly and unpredictably and can affect antibiotic treatment on a wide range of infections and diseases, including those that spread between animals and humans. So it's important to keep in mind that the same classes of antimicrobials are used in food animal production in treating our pets as they're used in human medicine. Now, there's some restrictions on that in food animal production. There are no restrictions on that in the antibiotics our veterinarians might use to treat infections in our companion animals. And so we know resistant bacteria can be found in food animals and food products destined for human consumption, and including those same genes and strain types as we know from illness in humans. And Stuart Levy, my mentor in my postdoc years, I thought put it just perfectly when he said antibiotics are societal drugs. It's the only class of essential medicines or any medicines which used in one environment can compromise or affect how effective they are used later on in a different environment. And so that brings with it a sort of new set of responsibilities, different ways of thinking, and One Health, which I'm sure you all are also familiar with, has been the framework in which WHO has tried to build capacity in countries to do surveillance for resistance and also to address it on the policy level. In the past, what we do in our NARMS program has been defined as integrated surveillance, and WHO defined that as the coordinated sampling and testing of bacteria from food animals, foods, and critically ill humans, and subsequent evaluation of resistance trends throughout the food production and supply chain using harmonized methods. And I'll show you how that's what we do in our NARMS program. But one thing that we've been contemplating that came out of both a recent review of the NARMS program and in our meeting today in the same place yesterday and the day before is what are the prospects and value in moving towards more of a One Health paradigm. And that One Health paradigm means sampling beyond that integrated food chain and looking into the environment and looking at animals and not just zoonotic bacteria from animals, but animal pathogens and elsewhere. And again, whole genome sequencing technology, especially metagenomics, is going to make this more possible than ever, I think. So the purpose of NARMS, what we've said publicly for many years, is to monitor trends in resistance, get this information to people who can act on it, conduct research to fill in the gaps. It's very difficult to sample yourself to every root cause or to every solution, and research has been an important part of filling in some of those gaps. More recently, it's become adopted by CDC to prioritize outbreaks based on now more than ever whole genome sequencing data, and it's an important part of FDA's regulatory processes whereby a new animal antibiotic being proposed for review by a sponsor goes through a series of qualitative risk assessment steps that include mechanisms of antibiotic resistance, co-resistance to other compounds, and so on. So in another way to look at this process overall and how surveillance fits into it, obviously surveillance is a key component to any public health action if you're going to get a hold of the magnitude and nature of whatever that hazard is, in this case resistance, and understand the benefit of any interventions that were targeted based on the baseline and trend data. So I describe NARMS as beginning with establishing baselines for resistance in different pathogens from different sources, how that resistance spreads, what it looks like over time, can we get to where it might be coming from so that our decision making, whether it's regulatory or not, is more of a scalpel than a hammer, as some describe that. Understanding the contribution or the relationship of antimicrobial use and resistance is a tough one we're still struggling with, but ultimately it hinges on human health impact and the burden of resistant infections in humans. And when that situation becomes untenable, then the interventions that are put in place, again, give NARMS, if we're doing our job right, an opportunity to measure that impact. Uh-oh. So this is the basic structure of the program. It is an interagency program between the FDA, the CDC, and the USDA. The Centers for Disease Control is getting isolates from the field, if you will, from clinical labs and physician labs around the country. Every 20th salmonella is subjected to standard in vitro antimicrobial susceptibility testing, but as we've heard, eventually all and soon all of the salmonella reported will also be sequenced. But we'll continue to have phenotypic data on 5% of them. USDA FSIS has a random sampling of national production at slaughter of the 4 major food animal species. And then at FDA, where the retail meat testing is done, in 2018, we'll have 22 states sampling 80 products every month of beef and pork and chicken and turkey, and then isolating salmonella, and then characterizing them phenotypically, and sequencing right now is being done at the FDA labs. So that's the integrated portion and structure right now. We combine these data into integrated reports where we try to harmonize our reporting as well as our methodology. And Our latest report, integrated report, came out this Monday. I think the CDC human report, which looks beyond Salmonella and Campylobacter to other enteric pathogens, should be coming out very soon. So the issue about how to, what does this mean for resistance monitoring, and beyond that, what does this mean for susceptibility testing, and maybe someday clinical care. You all know that currently what we do and what we've done for many decades to assess susceptibility in an organism is we do a biological response in serial dilutions of antibiotics and look for the lowest concentration that inhibits visible growth. There's a lot of talk about, well, how good is genomics going to be at bridging to these old methods? And I think I would start out by saying the old methods aren't as good as you might think they are. It's a biological response in an artificial situation, and similar to what Dr. Besser said about PFGE, it's a predictor of the likelihood of success when it's correlated with other parameters. But the method itself, the laboratory method itself, allows a 3-drug dilution range for QC for most organisms. Well, that could be 1 or 4 micrograms per mil. That's a pretty broad range. In some cases, CLSI permits a 4-drug dilution range to be in QC. So it's not, the chemists always would make fun of us when we tell them about this because it's very imprecise in a lot of ways. And it also doesn't always reflect all the things that go into resolving it doesn't reflect all that goes into resolving an infection. So I just want to begin by saying that while the gold standard is MIC testing for susceptibility, it's got its own perils, and so I think that's a good place to start. And so when we start looking at how well genomics can predict resistance or be useful in surveillance or in guiding clinical therapy, it's good to keep in mind that that's not a perfect method either. It doesn't always correlate well with clinical outcome as well. And in a lot of cases, the studies haven't been done at all. And so breakpoints will be borrowed from one pathogen or animal into another without the clinical outcome data. So you have this MIC. Well, what do you do with it? Well, the title of this is The Art and Science of Drawing a Line Somewhere, and it is an art and a science. And there's 2 ways right now in which the data are considered. One is how well does it predict clinical resistance? 1 MIC set with using a lot of data, using wild-type MIC distributions, using clinical outcome information, which is really the key one, using PK/PD data that addresses the concentration of the drug at the site of infection, and then standard of practice in medicine goes along with that, and FDA is responsible for setting those breakpoints. Another approach is what's called epidemiological cutoff values. Let's just identify everything that's no longer wild type and use that marker as a way to monitor resistance. And we could have a long conversation about fighting over what that word means, because to some it only means the former. It only means clinical resistance and clinical breakpoints, but in some cases to other people it means non-wild type or decreased susceptibility, depending on the questions you're asking. And so in one case here, the breakpoint for epidemiological cutoff values in this mock-up would be 0.25, or in this case 0.5. So here's the wild type distribution. You might have intermediate MICs in here. That used to be called indeterminate, recognizing the limitations of the method, and then a clinical breakpoint. And so where you set that point can affect the data that you are describing over time. Well, CLSI has tried to capture all these different definitions into one and describe resistant strains as those not inhibited by the usually achievable systemic concentrations of the agent with normal dosage schedules, and or fall in the range where specific resistance mechanisms are likely, and here's our genomics, or clinical efficacy has not been reliable in treatment schemes. So you can see they're trying to really make everyone happy, and sometimes the definition is as loose as the data. So you've all seen this slide in some iteration probably before, but when genomics became routine and affordable, the first question in our surveillance system is how do we incorporate it? How well can we bridge from the old to the new? How well does it predict resistance in pathogens? And we started off with a very basic question. What is the correlation between the presence of known resistance genes and isolates with an MIC above just the clinical breakpoint? And then I'll say a little bit about how we're exploring metagenomics as well. And we've done 3 studies, main studies looking at this, and as Dr. Goldman pointed out, the correlations for the bacteria under surveillance in NARMS is very high. 95 to 99% correlation between MICs above the clinical breakpoint and the presence of known resistance genes. And we've done it for Salmonella, Campylobacter, and E. coli. And Greg Tyson, who is here, has led a lot of this work and is working on Enterococcus. When NCBI and we started conversations about building these analytics into their pipelines, we expanded it to over 6,000 Salmonella now. The first study was 600. And the data hold up. It's very highly correlated with this one MIC, the resistant MIC, and the presence of known genes. We did a collaboration that's ongoing with Argonne National Labs where they took a machine learning approach and just said, can we predict MIC from the genome, blinded to the presence of any known resistance genes? And I couldn't explain the computer part of it. That's a little beyond me. But basically, it was a Kamer-based approach looking at correlation with one-fold serial dilution above or below the MIC. And again, it holds up incredibly well. This actually surprised me, and to see that you can actually predict all the MICs for which we had ranges with the same high degree of confidence. Now, I should have emphasized this work's ongoing, and we're still trying to get some more dilution ranges to improve the data. But it's incredible how well you can use the genomic data alone. So what CDC is doing in sequencing all the Salmonella is a resistance monitoring system in my mind because these correlations are so good. And so the data set has just taken off. With every Salmonella that's isolated, we can consider that in our risk assessment and regulatory processes at FDA because the correlation is solid. I'm going to skip just one slide. On the metagenomics side, just a few quick points. We have started to apply this technology both looking at animal cecal samples in NARMS and looking at the retail meat isolates to get an idea of really to sort of stress test the technology and explore its limitations for doing routine surveillance. And Daniel Tedeschi and our group has been working on this, and we have kept every cecal sample collected in NARMS for the last 3 years and have some 20,000 of them. Nationally representative randomized samples of U.S. food animal production. So we didn't want to let these samples go, in part because new policy changes were coming, and so we've saved them all and have started to look at them metagenomically and started, as this slide shows, to associate specific resistance alleles with different animal sources, and also in the retail meat samples as well. So stay tuned for updates on that. In some of our earliest studies were back in, oh my goodness, it must have been in 05, 06 when we did our first sequencing, and it still was very expensive. We got some information that I think is still a very good illustration of what the data mean to us and different ways in which it can be presented. And this plasmid just happens to show one of our first outputs, which was a multi-drug resistant Salmonella Newport on a backbone that was essentially identical to a strain from a child in Madagascar who had plague. So this plasmid, when you start thinking about One Health, you see the importance of global One Health. What does it mean? Well, it certainly means these things can develop quickly and spread rapidly around the world and become, and what is seen on halfway around the world can become fairly common in the U.S. food supply. And this just shows different arrangements of genes. But another part of it that's interesting and important is we see associations with decontamination, resistance to decontaminating chemicals used in processing plants. So new drivers of resistance emerge in these data, new associations and arrangements of genes with some indication of where they might be around the world. And the next event is here. The next slide just shows a spike in gentamicin resistance that we saw in NARMS data, and it just illustrates the fact that we'd have never gotten to the bottom of the genes behind this without genomics because the PCR primers available for aminoglycoside resistance genes at that time didn't have all the alleles we found. So the new discovery is obviously an obvious part of what we get from whole genome sequence data routinely now. So what we called research and ad hoc PCR studies is now routine surveillance. Another important event that showed the value of this technology was plasmid-mediated colistin resistance that emerged in China in the fall of 2015. And Errol Strain was really helpful in this, in going into NCBI's database and saying, have we ever seen this in a domestic isolate of any bacterium in the United States? And at that time, screened 155,000 genomes at NCBI and said it wasn't there. Incredibly powerful to do retrospective surveillance like this without opening the freezer. We later did studies of selective enrichment and found MCR1 in 2 out of 500 swine samples, and the metagenomics tools we were developing at the time picked it up as well. So that bodes well for the future of metagenomic component to the surveillance. That's just a few illustrations of how we're building it into NARMS. I would invite you to go look at our last report that came out Monday, where we've put genomics now into our data set. We went back and sequenced every retail meat, salmonella back to 2002, and as David Goldman pointed out, they're sequencing all the animal isolates. Next year we'll have all the human isolates. So knowing how good the predictions were of genotype to phenotype, we put quite a bit of effort into interactive data displays where you can look at resistance over time and see the genes that go with it from the different sources. And it's really powerful, and it gives people access to the data in new ways and allows them to ask and answer questions. One of the good ones is we've gotten beyond this really crude metric of MDR, multi-drug resistance, being resistance to 3 or more drugs. Well, what drugs, right? So now you can go in and see what drugs and refine the analysis and have more confidence in what you're talking about in the trend. Another thing we've developed is to take the next obvious step. We harvested all the NCBI data, screened it for all its resistance genes, and took advantage of whatever metadata that were there with its strengths and limitations. And you can do the same thing now in this tool we call Resistome Tracker, where you can look in any source according to the metadata categories at any resistance gene, in this case aminoglycoside resistance gene AADA1 in chicken. You can see where it is at NCBI by biosample, and then what our intention was just to sit it on top of NCBI. You can click on any of those isolates with any of the genes you're interested in and go right to the SNP tree. So this just seemed like an obvious thing to do based on NARMS data and taking advantage of the sequencing that was coming out. So, and then the last tool we built into Resistome Tracker is, well, what's new? We want to know the wallpaper in the room, if you will. What's the backdrop for analyzing our domestic data in NARMS? So any new gene that comes up in any submission to NCBI, we've put an alert system on there so we can track it and see the data in which it was reported. And Heather Tate, who is here, has developed this Resistome Tracker tool, and we're going to try to launch it. It's not out yet. We got busy this week, but maybe in the next week or two, you'll be able to play around with this and see if you like it. It includes a mapping tool where you can look at over time when these different resistance genes appeared around the world, and you can apply this, of course, to any pathogen. So we've put a lot of effort into making sense of the complex data that we are all generating through genomics. So in closing, One Health, One Method, I know it's a bit of an exaggeration, but it sure feels like it. We can predict so much from the genome. We can predict resistance in our target pathogens so well from the sequence data. It overcomes so many past limitations with the metagenomics, our reliance on cultivatable organisms, which is expensive and obviously very narrow in what we can say about the resistome. The best part to me, the second bullet, it permits us to look farther with fewer resources and lower costs. So now we can get out and look at the coastal waterways and look at surface water systems and start incorporating companion animal and other testing because we can glean what we need from these samples. And that's where we're trying to go next. I've noted how it reveals new associations, including determinants, perhaps of animal origin, of other drivers of resistance and new alleles. Greater confidence in decision making, that's important. We would like to know not just where a pathogen comes from, but where a resistance comes from. And with that, I can't thank everyone, but it's been a tremendous opportunity to collaborate broadly, and it's been a real satisfying experience working with NCBI, who we'll hear from next, and CIFSAN and CDC and USDA on this. Thank you.
So this is great. Everyone's talked about NCBI, so I don't have much to talk about. Go to the next slide, please. So those of you not familiar with what NCBI is, we are the premier biomedical informatics institute on the planet. We are the host for PubMed, all the medical literature, over 23 million publications, linked to all the clinical data, and all the sequence data that you've heard about from today is being submitted to our databases. And then we have specialized databases as tools, and I'll talk about one in particular today called our pathogen detection system. Next slide. And again, to reiterate what someone has already said, we share all the sequence data with the European Bioinformatics Institute and the DNA database of Japan within 24 hours of the data being released. Next slide, please. Go to the next one. This is a rough schematic of how our system for the pathogen detection pipeline works. We have submitters on the left, including FDA Genome Tracker and CDC PulseNet and USDA, submitting data to us into the public databases. And then we have a pipeline, which I won't go into a lot of detail today. There'll be one other slide on that. We basically do an assembly, clustering, and produce phylogenetic trees, and we make the reports publicly available to all of you. Next slide, please. I want to talk briefly about the metadata. This is the template that we built, and you've seen that from a few other people already this morning. This came about from discussions with FDA about the sort of metadata they wanted to see submitted to the databases that would enable them to do their job. It's basically 4 categories of information. What is the sample, including the organism, a unique identifier such as a strain or isolate number. It's categorization, whether it comes from a clinical host-associated sample or from a food or environmental sample, and then a few other categories such as when, where, and who. The ones I've colored in yellow are the absolutely minimal sample fields that need to be filled out in order to submit using this template, and that's what CDC is using right now. And then 6 months after the sequence gets deposited, they update with some additional metadata fields. For USDA and FDA, and you've already heard from some of them this morning, they are willing to submit things like the geographic location, the year or month that the data was submitted as well. This has been an incredibly successful template. So we have many, many other templates for bacteria, for example, at NCBI. This one has been used by over 256,000 submissions to date. And that's not just samples from PulseNet and Genome Tracker. That includes other academic laboratories and other state actors that are willing to submit data to us. And you can see the breakdown of the clinical versus the food and environmental. I'll talk about some of these metadata fields later on. Next slide. So basically what we're trying to do in this system is we're taking very large volumes of data and reducing it to relevant data. So we might have hundreds of millions of base pairs in a short read sequence for one particular isolate. We do an assembly, and then we're turning that around into a phylogenetic tree along with associated antimicrobial resistance genes. And we want to do that within 24 hours of the sequence being deposited. So turning big data into useful data. Next slide, please. And we have to do this because we know that the sequences that are coming in are going to increase in the future. So this is just a snapshot of the last 6 months of the data that's been submitted to our pathogen detection system. And you can see it's dominated by Salmonella in that orange slice of the pie. And then the other 3 foodborne pathogens are basically making up the other 95% of that. And so we have a few clinical things like Klebsiella coming in, but basically it's the foodborne isolates that are coming into our system. And we know that the Salmonella that are being collected in the U.S. should all be sequenced by the end of 2018. And so actually we should see the rate of that double within the next 18 months or so. So we should expect, I believe it's 90,000 isolates per year for all the foodborne pathogens in the U.S. And you can see at the bottom is the graph showing the sort of number of submissions per day. And you can see we first turned this on in 2013. That's when we first started the real-time Listeria project, where all Listeria in the U.S. are being sequenced and submitted to us, and that's ramping up for the other foodborne pathogens. But you can see both the spiky nature. Sometimes we get large volumes of submissions from, Public Health England is an international partner that sometimes submits in batch mode. They might submit 1,000 or 10,000 in a day sometimes. But you can see it's growing, and we would expect that rate to increase as we move forward into the future. Next slide, please. This was a snapshot of data I presented at NIST as part of their standards for pathogen detection. This was basically looking at the total number of submissions and whether they're clustered or not for the 4 foodborne pathogens. And again, you can see Salmonella is predominant on this slide. The interesting thing about the clustering, so I didn't mention this, and I'll probably mention it in the next slide or so. We make clusters of 50 SNPs or less. So very closely related isolates. And you can see most of Listeria and most of Salmonella are clustered. E. coli and Shigella not so much. So that's an important byproduct of the biology of those organisms. And I'll come back to this in some of the analysis I do. Next slide, please. So this is the last pipeline slide you'll see. So those of you who are at my other talks can breathe a sigh of relief. You don't need to see any more of those. But basically, the data comes into us, and it's several days before it gets sequenced and submitted to NCBI. But we do an assembly, and we are starting to do WGMLSD allele calling and producing a table of nearest neighbors. We're doing that now for Salmonella and Listeria, and that's been running for 2.5 months. And we're aiming to get that data back to FDA within 1 hour of the sequence being deposited. So that's an incredibly fast turnaround time. When the sequence comes off the machine, comes to NCBI, and if Errol does it right, he says, he'll submit it in the morning, have his coffee, and then he can look at the results within, by lunchtime. And he can make a decision about inclusion, exclusion within that 1 hour. We're also doing SNP clustering. This slide, we're planning to replace the initial clustering we do using whole genome MST, and we aim to get the clusters of these nearest neighbors and into phylogenetic trees and on the web-based interface, which I'll show you some examples of within 24 hours. So the rapid reports just gives them an extra day to say, these isolates are related or not related, and make decisions based on that. Next slide, please. So just for the rapid reports, like I said, we're doing Salmonella and Listeria. We're reporting the 5 nearest neighbors and all neighbors with less than 6 allele differences, and that cutoff may change. We're right in the pilot phase of this project, and we've already gotten some preliminary feedback that we may want to change that threshold. We're reporting the number of differences, number in common, and the SNP accession if it exists. So if they can actually see within the list of nearest neighbors that some of those are already participants in an existing SNP cluster, they can actually take additional steps. And we put this into 1 file per run, and those reports are already being made available on the FDP site. So I won't show you an example of that, but you can go take a look. Next slide, please. And all this data is being made available into our publicly available pathogen detection website. So you can go to this page right now and see all the data being submitted from Genome Tracker and PulseNet, and you can interrogate the data, and I'll show you some examples of that. There's a couple ways into it, but we already have over 174,000 pathogens and at least 142,000 with either an acquired or chromosomal antibiotic resistance gene. Next slide. So I just wanted to do a quick analysis, just basically for my own benefit. Using the metadata that's been submitted, I wanted to look at the isolation source. So the isolation source is the definition of that is it describes the physical, environmental, and or local geographical source of the biological sample from which it was derived. So for a clinical isolate, that might be blood, stool, urine. For a food or environmental isolate, it could be a riverbed, it could be a food contact surface, it could be an environmental swab, it could be cheese. So next slide. So I simply wanted to ask the question, if I look at all of the submissions coming into our system from CDC, FDA, and USDA, including the state labs that are submitting under those Genome Tracker and PulseNet submissions, and if I want to look at all pairs within 10 SNPs, what am I going to see? And I want to categorize each one of those pairs, either they're clinical versus food or environmental, and that could be potentially the smoking gun for causing the clinical illness, and then food and environmental versus food and environmental. So go to the next slide. So this is a summary of those pairs. We're just looking at the counts. We have the 4 foodborne pathogens at the bottom, so Campylobacter, E. coli, Shigella, Listeria, and Salmonella. And then the pairs are those 3 categories I just told you about. Clinical are red, clinical environmental are green, and environmental are blue. So you can see, obviously, Salmonella clinical cases completely dominates this slide. I'm not telling you anything new. You already knew that Salmonella is one of the biggest problems in the U.S. What you will see, though, is that the number of cases of those pairs is not uniformly distributed. So Campylobacter is pretty uniform between the 3 categories. E. coli, Shigella, and Listeria, it's not. And so I think this, again, informs you about something about the biology of those pairs and the distances that I use. I use a 10 SNP threshold. That was an arbitrary pick. A lot of people have come to these meetings and said, what is your threshold? You can't, again, I'll reiterate, you cannot simply just pick one threshold and get the answer. There's additional contextual information. Go to the next slide. If I look at the number of samples within those thresholds, you can see most of the samples in Salmonella submitted are within that 10 SNP threshold to something else. That's not the same for Listeria. About 1/3 of the clinical cases are within that SNP threshold. And so it varies, again, between all the organisms. The last column is also interesting because I said we make these SNP clusters of 50 SNPs or less. There's a number of cases that are just not clustered with anything or not clustered with a food or environmental isolate. So there's actually over 2,000 clinical Salmonella not linked to any food or environmental isolate at all in our database. and 1,500 E. coli, and I'll come back to that just near the end of my talk. Next slide. So these next 4 slides are just looking at the 4 foodborne pathogens and what that isolation source breakdown is. And this is free text, so I'm basically just doing a naive sort of smushing together of some of the terms into these sort of categories. So we don't have 100 rows, we have only 20 rows or so. And again, you can see salmonella, predominantly chicken. You have some beef, pork, water. You have papaya, and it's highlighted in yellow, and I'll come back to that. And then things like a research strain, so somebody in Micro 101 is just not doing, the proper technique that they're supposed to be learning. Next slide. Listeria, again, swabs, environmental swabs, cheese, and then various food products. Next slide, please. And then E. coli, Shigella, dominated by soy nut butter, and then beef, milk, goat, etc. Next slide. And then Campylobacter, of course, milk is a big problem, unpasteurized milk, and then beef again. So go to the next slide. And this, of course, completely matches what you see on the multi-steroid outbreaks for CDC. You see soy nut butter for E. coli, you see papaya. There's a huge papaya problem from Mexico, and you see cheese as a problem for Listeria. And so I'm not trying to present this as like a detailed scientific analysis of the metadata. I'm just simply saying that we make all this data available. You can do these types of analysis. This only took me a couple hours in an afternoon. You could do similar sorts of things, and I'll dive into some more detailed analysis that you can do. So next slide, please. So for example, a lot of people talked about our isolates browser. Here I'm just doing a search for papaya into our isolates browser. You probably can't see it in the back, but there's 225 isolates that come back with a search term of papaya in our existing database when I did this. That big table in the back is called the isolate browser, and basically every row in that table is an assembled genome that's come through our system or come through GenBank. It's got the associated metadata, and you can actually, add columns to that based on what you want to do. We do a separate thing where we intersect the number of search terms with the number of clusters, and that's a little table at the bottom, which is actually on the default page on the upper left. If you can't read that, it says, at the top row, 23 of the total 32 isolates in this cluster have the search term papaya. If you go to the next slide. So this is our new tree viewer, which is not available to you. This is being alpha tested by FDA right now. And so if you went to our page and clicked on one of the SNP trees and you saw some examples of that earlier, you wouldn't see this exact page. But this is something we're testing with the FDA because we want to make tools available to them that will help them make a decision based on genetic distance. So this is one example. This is a cluster of 710 isolates. 8 of them have the search term papaya. But only one of them is from 2017. If you see on the left, there's a breakdown by the date. And if you look at that part of the tree where that food and environmental isolate hits, it's basically on a completely separate branch of the tree. It's not near any clinical isolates. It's at least over 30 SNPs away from any clinical case. And so that particular papaya probably is not a causative of any clinical illness that's been recorded. If we go to the next slide, though, we see the opposite. So this is another salmonella cluster. It's got over 1,100 isolates. 8 of them, again, are from papaya, but 3 of them are from 2017. And if we look at the SNP tree, you can see one environmental isolate is incredibly close to a large number of clinical isolates. Go to the next slide. And if you look at the entire subtree, you'll see there's 112 isolates in total, and they're incredibly closely related. The max SNP distance is 17. The average SNP distance is 3. If we go to the next slide. If we just look at the food and environmental isolates in that total list of 112, 3 of them exist. One is that papaya from Mexico, and 2 are papayas from the U.S. And so what I showed you is I think that you can quickly see whether something is inclusive or exclusive. Make a decision very quickly using just the genetic distance with some limited metadata, and then you can go on to the rest of whatever your job is, whether you're a public health or a regulatory agency. And again, so we're building tools to facilitate that. We're not the agency that makes those decisions, but we want to make tools that get you 95% of the way there without any extra effort. Next slide. So just going back to those unlinked isolates, again, clinical isolates not within 50 SNPs of any food or environmental isolate. They are not just singletons. I thought when I first did this last week that these would all be completely unlinked to anything, but it's not true. Some of them are actually in some very large clusters. So my question, and I'm going to ask these questions, I don't know the answer to them. Do we need a new sampling strategy? Are there new food vehicles that are waiting to be discovered? What does the epidemiology tell us? And I can't answer those questions because I don't have that information. You people have that information, and so you should be asking these questions and be able to answer them. again, pointing out the number of isolates. And if we go to one next slide, we just look at one of those. It's an extremely large SE cluster of 506 clinical cases. No, there's no food or environmental isolates in this cluster at all. They're all collected from 2013 to 2017. They come from the U.S. They come from Public Health England, so there's isolates in the UK that also cluster with these isolates. They come from state labs. So what is the cause? I've heard something, I've heard a rumor, but I'm not going to tell you because I don't think I can share that. But obviously, these are the sort of things we would need to be looking at, based on the questions I asked earlier. Using our interface, you can do that, but also the places that produce this data should be asking these questions and answering them. Next slide. Just to basically end on the antibiotic resistance, you've already seen some of the talks using some of our databases and some of the analysis that we're doing. This came about because the president at the time decided we needed to combat antibiotic resistance, and so he put in motion this thing called the CARB report, from the president's scientific committee, combating antibiotic resistance. And NIH is mentioned twice in there, at least for two critical elements. One is to produce a reference database, a well-curated reference database, and maintain a national sequence database of resistant pathogens. So go to the next slide. So we've done that in a number of ways. First, we've actually built a template to capture the susceptibility data that people are collecting. It's called an antibiogram. I guess in addition to the sample metadata, the minimal template that we're collecting, you can actually submit AST data as well. We have almost 5,000 submissions using that template right now. And we have put together a reference database of antibiotic resistance genes. Now, this is not something we just did on our own. There's a lot of people out there who say, well, which database should I use? Well, we have a collaboration going with CARD. We may have a collaboration with ResFinder, depending on some upcoming discussions we have. We've taken over the LAHI database. So they were the place that you would submit novel beta-lactamase alleles to, and they would assign a new novel beta-lactamase allele, let's say SHV or TEM. They're retiring, and so they've asked us to take over that responsibility. And so we're the place where you would actually submit those for, once you want to make a publication into JAC or AAC, you would actually submit to us, get the novel allele number, and then goes into your publication. Once this gets released, it gets fed into our reference database and used. And so we're implementing tools for identifying those genes using that reference database. When we go to the next slide, we're actually integrating that back into the isolate browser that I just showed you, so you can actually see the list of genes per isolate integrated directly into this. So this is an example of another large salmonella cluster. We have 15 isolates that encode an MCR, so a mobile call stem resistance gene. And you can see just one example, probably you can't see, but there's a cluster of 4 isolates from Thailand that all have an MCR1 gene. So this system will allow you to interrogate for not only metadata, but also the presence-absence of different resistance genes. Next slide. So just to summarize, we're enhancing our existing analytical pipelines to improve the turnaround time to answer, basically these two fundamental questions. Are these isolates clonally related? Is there a point source for clinical illnesses? And we're improving these interfaces, enhancing the information that's layered on top of them, including antibiotic resistance genes, making the system much more easy for you to use to make determination of inclusion-exclusion. And then in the future, we'll be adding things like virulence genes, heavy metal resistance, point mutations, and mobile elements. Next slide. So that's it. If you have any questions, you can email that email address or come see me here. This is all the people that have worked on the pipeline at the various points. So you can see it's basically an army of people at NCBI that have been involved in this. Although the day-to-day operations, the number of people involved are actually just a few people. I'd like to thank all of our colleagues at FDA, CDC, and USDA for making the data available, because we wouldn't be able to build these tools on top of that if they didn't make the data available. And thanks to David Littman for helping to push this system out, even though he's now gone off to work on food products, actually. And I'll be happy to take any questions at any point.
Thanks, Bill. So we're going to go for a quick coffee break, and we'll be back here at 3 o'clock with 2 additional presentations and then opportunity for questions and answers.